Hello guys, a very warm welcome to all of you. Today in this video, we'll be talking about JKSET examination paper, which was recently conducted by JKSET agency. Okay. So in this video, we'll be discussing the questions and try to answer all of the questions of life science subject. So starting with the question number one, terrestrial vertebrates evolved from fish in which period? So it is Devonian period because this is also called as age of fish. Okay, remember, don't say fishes. Now the question number two. DNA double helix is stabilized by hydrogen bonds and base stacking interactions. Question number five. Which of the following is not involved in post-transcriptional control of gene regulation? Option A is riboswitches. Yes, they are involved, as you have seen in tryptophan operon, where that hairpin loops between different leader sequences bring about either activation or or suppression of the gene expression. Riboswitches, yes. DNA methylation, no, this is not involved. So DNA methylation is the correct answer here. Okay. Yes, microRNA is also involved. So is RNA interference. Question number six. Which of the following is the main site for lipid biosynthesis? It is smooth endoplasmic reticulum. Okay. Option D. Now, question number three. Isoelectric point is the pH at which protein has more positive charge, no. More negative charge, no. Neither positive nor negative charge. Uh, no, it's not correct because it has positive as well as negative charges as NH4 and COO negative. Okay, so D, because these positive and negative charges are equal at isoelectric point. Isoelectric point is the pH at which protein has equal positive and negative charges. Okay. Question number four. Nicotinamide adenine dinucleotide phosphate is generated in. It is generated in pentose phosphate pathway. It is also generated in glycolysis, but in glycolysis there is NADH. Okay, there is no NADP. Mhm. Okay, so question number four. Now, question number seven is following is the list of enzymes that act on different intermediate products of glycolysis. Okay, so here is a list of enzymes involved in glycolysis. What you have to do, you have to arrange them in the correct sequence according to the steps in the glycolysis. Okay, so the first enzyme that we know is involved in hexokinase, which changes glucose to glucose 6 phosphate. Okay, two. So you have option C or option D here. Okay, and then four. Yes, phosphoglucoisomerase, which changes this glucose 6 phosphate into fructose 6 phosphate. Fructose 6 phosphate. Then this fructose 6 phosphate is again phosphorylated by phosphofructokinase. Phosphofructokinase. So, okay, hexokinase is the first. Second is this. This is third, and finally we have this. So we have 2 4 3 1 2 4 3 1. So option C is the correct answer. Moving on to the question number 8. Present F1 particles present in the mitochondria are located in they are located in inner surface of inner membrane of mitochondria. Which combination? Question number 9. Which combination of cyclins and CDKs work together for a cell to pass from G2 to M checkpoint? Cyclin A, CDK. No, they are not. Cyclin, yes. Cyclin B, CDK1, or the cyclin CDK protein is involved in G2 to M checkpoint. Cyclin A is involved in synthesis phase, S phase. They are S phase cyclins. Okay. Which question number 10? Question number 10 is which one of the following cellular organelle is affected in Zellweger syndrome? It's a peroxisomal syndrome. Yes. In the mismatch repair system, question number 11 is in the mismatch repair system in E. coli. Mute S, mute H, and mute L are the key players. Yes, the repair of mismatch in the newly replicated DNA can be initiated by these proteins because mute S recognizes the mismatch and activates the endonuclease. Mute H, it is somehow true because mute S recognizes the mismatch. Yes, and activates the endonuclease. Yes, is involved in activation, but along with mute L. So, what about option B? Mute S recognizes the mismatch and binds as a complex with mute L and mute H. No, it binds with mute L, and then they both recruit mute H. So, option C is the adenine is methylated in the newly synthesized strand. No, the complex recognizes unmethylated adenine in the GATC sequences in newly synthesized strand. This is also correct. So is this. I think. I think I will go with. I think I will go with one. Okay. I think I will go with the option A. Option four could also be correct. Let's see what comes in the answer key. So, question number 12: which of the following represents the character equation for Spearman's rank correlation coefficient? It is rho is equal to this. It's option A. Now, question number 13. Question number 13 is which one of the following statements is not correct? Mitochondria release cytochrome C into the cytosol and triggers programmed cell death through apoptosis. This is correct. In biological membranes, integral proteins and lipids interact mainly by hydrophobic interactions. Correct. All membrane processes such as pumping and channeling of molecules are carried out by proteins. Absolutely. Channel proteins can facilitate both active and passive transfer. No, channel proteins are passive. So option D is the correct option. Okay. Question number 14. In size exclusion chromatography, the smallest molecule is eluted. This is this is a somewhat confusing question. So, in the initial fractions of protein, no. In the last fractions of protein, yes. Smallest molecule not eluted. Smallest molecule comes along. No. So, in the last fractions of the protein is the right answer, because in case of size exclusion chromatography or gel exclusion chromatography, the smallest molecule or the smaller molecules go through the pores that are present in the matrix, while as the larger molecules they do not go into the pores and they directly come out okay of the chromatography apparatus. Okay, so question number 15. Which one of the following cell organelle and its function is mismatched? Lysosome is the breakdown digestion. This is correct. Peroxisome is transporting, modifying, and packaging proteins. No, this is done by ER and Golgi. So option B for 15. Cytoskeleton responsible for locomotion. This is correct. Centrosome major role in organizing the microtubule. Yes, this is also correct. Question number 16 is the development of pesticides. Question number 16. The development of pesticide resistance in insects. DDT resistance mosquitoes, for example, is an example of, okay, obviously it's natural selection. Okay, question number 17. The following graphs represent beta galactosidase activity in E. coli culture at different points of time before and after addition of lactose. Okay, beta lactose, which is beta galactosidase, which is a part of the lag operon, and lag operon is an inducible operon and is both positively controlled and negatively controlled. So here in this case, which one of the following graphs is a correct representation of activity of a mutant? It's a mutant here that does not synthesize the lag repressor protein. So here the lag repressor protein is not synthesized, so it will be, so the operon will be on, and galactosidase activity will always be there. So which one of these graphs represent the correct one? Okay, I think option D is the correct, because it is constitutively expressed whether whenever you add lactose it would be expressing itself because there is no lag repressor protein. Okay, moving on to question number 18. In question number 18, in bacteria, row independent termination of transcription involves. Row independent. That means the protein which is involved in termination of transcription, okay, is not involved here. So what happens here? A stem loop structure formed in the transcript RNA molecule, which pulls out the RNA molecule from the RNA polymerase complex. Okay, question number 19. Match list 1 and list 2 and select the correct answer from the codes given below. Okay, so Nokrak is present in, I'm not sure. Sundarbans in West Bengal, yes. So Q1, I think I got the answer. Q1, yes. So A is the correct answer here. P3, Nokrek in Meghalaya, or 5, okay, or 5 in Madhya Pradesh, Pana in Madhya Pradesh, Pana Biosphere Reserve, okay, Gulf of Mannar, S4, Tamil Nadu, Nanda Devi, okay, where is Nanda Devi? It's in Uttarakhand. Okay, question number 20. So the question number 20 says eukaryotic RNA polymerase third transcribes tRNA, right? Yes, it also transcribes 5S RNA. And 18S no, microRNA, messenger RNA no, 20S RNA no. So 1 and 2 only. Option A. Question number 21. In bacteria, a sequence called dash sequence is important for formation of translation. Sorry, initiation of translation. This sequence base pairs with, in bacteria, which sequence is important for initiation of translation? It's Shine-Dalgarno sequence. Shine-Dalgarno sequence. Okay, Kozak sequence is in eukaryotes. Okay, this sequence base pairs with dash and end dash end of the dash rRNA. Of in eukaryotes we have, okay, initiation is done by the smaller ribosomal subunit which contains 16S RNA. Okay, so option B is the correct at the 3' end. Yes, right. In the DNA segment, question number 22. In the DNA segment given in diagram below, the replication fork moves from left to right. Okay, so are the two strands. The replication fork goes from here to here. Okay, based on the diagram, the following statements were made. Strand A is the leading strand. Strand A is the leading strand. No, because the replication fork goes from here to here, and its synthesis will be from this end to this end in patches. So this is not a leading strand. No, one is not right. Strand B is the lagging strand. Oh, not at all. This is the leading strand. Okazaki fragments will be formed on strand A, right. Okazaki fragments will be formed on strand A, right. So which of the following statements will present correct set? Okay, only three. Option A is the correct one. Going to question number 23. Consider the following statements. Human papillomavirus infection is a well-established cause of cervical cancer. Yes, this statement is correct. Everybody knows it. Statement 2 is bivalent and quadrivalent vaccines against HPV were licensed in India in 2008. This is also correct because I looked it up from the internet. Yes. Statement 3 says Gardasil and Cervavac are the quadrivalent vaccines against HPV. Yes. India's first indigenously made HPV vaccine, Gardasil. This is not correct because India's first indigenously made HPV vaccine is this guy. Was launched by Serum Institute. Okay, this was launched of obviously this was launched by Serum Institute of India. Quadrivalent is which protects us against 4 kinds of HPVs, and bivalent is which protects us against 2 types of HPVs. So option statement 1, 2, and 3 are correct. 4 is wrong. So only 4. Option D is the correct answer here. Question number 24. The effect of neurotransmitter acetylcholine on the rapid relaxation of smooth muscle cells is signaled via its GPCR. Okay, it is G protein coupled receptor. Cell signaling pathway not regulated by ubiquitination. This is a tough question. Okay, I'm not sure yet. Let's come back to this question in the last part of this video. So, okay, Burkitt's lymphoma is a cancer of B lymphocytes. This is B for B. Which of the following apply uniquely to secondary lymphoid organs? Presence of precursor B and T cells. No, this is present in primary lymphoid organs. Circulation of lymphocytes. Um, Lymphocytes are activated. Yes, this is correct because in the secondary lymphoid organs, the lymphocytes are exposed to the antigens and they get activated. Question number 28. Which of the following is incorrect about the processing of an antigen such as bacterial protein in the acid compartments of the cell? Acid compartments of the cell are lysosomes. Lysosomes, okay, endosomes. It results in the production of potentially immunogenic peptides that associate with MHC class second. Yes, this is right. It may lead to activation of CD4T. This is also right. It may lead to activation of no. So this is wrong. We have to go for incorrect. So it may lead to the activation of CD8 T cells. This is wrong. Okay. Question number 29. Which of the following is true for direct allo recognition? Direct allo recognition, and you have indirect allo recognition. Allo means a foreign, foreign particle, foreign antigen recognition. Donor dendritic cells in the allograft migrate to secondary lymphoid tissues where they present allogenic MSC molecules to host T cells. Yes, this is correct. Okay, so we don't need to go for the other three statements. Okay, moving on to question number 30. In a biochemical pathway, if the product of gene A is required for gene B to function, gene A is said to be is required. I think epistatic to gene B. Yes. What is novum? Okay, sorry. What is nomen novum? A new species description, a replacement name for preoccupied taxon. Yes, correct. There is that nomen nudum. Nomen nudum. Okay, which is equivalent to a naked name. That's a name of a taxa where you do not have adequate description of that very taxa, and that's why it's called nomen nudum, because it's stripped of its description. Okay, so question number 32. Mm, which of the following correctly represents the conversion process in the nitrogen cycle? Okay, ammonification conversion. Okay, ammonification is conversion of organic nitrogen to NH3. Okay, yes, I think it's right. Nitrification is conversion of nitrate to ammonia. No, not at all. Nitrification is conversion of ammonia to nitrate. Denitrification is conversion of nitrogen gas. No, it is conversion into nitrogen gas. So no, no. Option D, nitrogen fixation, conversion of nitrate to nitrogen. No, it's actually conversion of nitrogen gas to ammonia. Okay. Question number 33. Following are some of the characteristics of restriction endonucleases. Okay. So first characteristic is nature of the enzyme, which is bifunctional enzyme with both endonuclease and methylase activity. Right. Protein structure, three identical subunits. Yes. Type 1 has three identical subunits. Yes, this could be true. Requirement for restriction activity needs ATP and magnesium 2 positive. Yes, type 1 and type 2 needs ATP, so this could also be right. And 4 is cleavage site at or near restriction site. This is also correct. But for one, okay, select the correct option for type 1 restriction endonuclease. Type 1. So for type 1, it's a bifunctional enzyme. Yes, it has three identical subunits, right? Requirement of restriction activity needs ATP. Yes. Cleavage site, no, it's not precise. Type 1 is not precise because the cleavage site is random for type 1. So it's random and it is away from the recognition site. Uh, so select the correct option for type 1 restriction endonucleases. So 1 and 2 is the correct option. Okay. Going to question number 34. Question number 34 says match list 1 and list 2 and select the correct answer from the codes given below. So you have two lists and you have to match these techniques with their utilization. So thermal imaging. Thermal imaging is used in, okay, heat loss. So heat loss means it is related to thermal. So P3. Okay. Hyperspectral imaging is a kind of remote sensing technique which uses wide range of wavelengths. So yes, Q4. Okay. Okay, data across, capturing data across wide range of wavelengths. So Q4. Then you have SAR, synthetic aperture radar. I'm not sure about this. And LiDAR, light detection and ranging. I think it's used for 3D mapping. Okay, S2. So and R1. Okay, so where is the, okay, it's below. So the options are A, B, C, D. Now, P3 Q4. P3, okay, P3 Q4. So option C should be the correct answer here. Okay. Yes. Now, going to the next question, which is question number 35. According to this question, which of the following is the correct order of steps in the ELISA process? ELISA is enzyme linked immunosorbent assay. It is used for detection of various antigens. So the first step in ELISA is coating the plate. So here it starts with blocking. No, it starts with B. Could be the correct answer. So coating the plate, then blocking. Yes, and then you add the sample, then you add the enzyme conjugated with detection antibody. Yes, substrate addition, stopping the reaction, and measuring the results. Right. Yes, this is, I think, the correct sequence. What about C? No, it directly adds the sample. In case of D, coating this palate, adding the enzyme. No. So option B is the correct sequence of steps involved in ELISA. Question number 35 is done. Moving on to question number 36. What does it say? Match the hormone action of 1,2,5 dihydroxycholecalciferol, which is active form of vitamin D, with its corresponding effects or characteristics. I'm not sure about this. We will get back to this question later. Okay. Um, now, question number 37 is, what clinical application is circular dichroism spectroscopy commonly used for? It is used for detecting the changes in secondary structures of proteins. So, drug discovery and metabolomics, no, not much. Monitoring protein misfolding in diseases like Alzheimer's, yes, it could be used for this. And option C is measuring oxidative stress in tissues, no. Investigating protein ligand binding affinities. This could be, yes, this could also be done, but I think in case of clinical uses, option B is the correct one. Yes, option B is the correct. So, question number 38. A person receiving an injection of gamma globulin as a protection against hepatitis is an example of it's an example of protein, sorry, a passive immunity, and it's artificially acquired. Yes, option C. In sea urchins and mammals, a rise in calcium 2 positive concentration following fertilization is responsible for cortical granule reaction. The source of calcium 2 positive ions is, it's yes, it comes from the egg itself. So calcium sequestered in organelles within the egg in both sea urchins and mammals. So 39's answer is B. Okay. Question number 40. Which PET radio tracer is commonly used to assess brain activity and metabolic rates? It's this FDG, fluorodeoxyglucose. During question number 41, during amphibian development, the localization of beta catenin in dorsal nuclei is an important event in establishing the dorsoventral polarity, which of the following proteins contributes to the above localization. It is this dishelved protein. Okay. Okay. 42. Question number 42. The first binary decision in mammalian development is to form primary germ cells or trophoblasts. No, trophoblasts are inner mast cell. Yes, trophoblasts then form the suspensor. Okay, so question number 42, 43. Question number 43 is, the technique used to study the dynamics and mobility of fluorescently labeled molecules within the living cell is known as fluorescent microscopy. No, LSCMS, FACS. It's FRAP, fluorescence recovery after photo bleaching. Okay. Uh, okay. Hans Sperman and Hilde Mangold identified the new Coop Center, demonstrated that blastophore lip of amphibian functioned as the yes. As they grafted it on the ventral side, that ventral side became the dorsal side. So, okay. Mm, Question number 45. The clavata 3 Wuschel signaling pathway has evolved as the central regulatory pathway that coordinates stem cell proliferation with differentiation in this maintenance of erection. Okay, option A, that maintains the amount of stem cells and also coordinates differentiation. So it's kind of a feedback loop that regulates this process. Question number 46. Which of these animals is an example of eusociality? So it's a famous naked mole rat. The fossil of Lucy belongs to which species of early human ancestors? Sapiens, Erectus, Habilis? No, it is this Australopithecus afarensis. Question number 48. Victor Ambrose and Gary Raukin awarded Nobel Prize in Physiology and Medicine for the discovery of microRNA. 24's answer is A option. Given below are two statements. 49. Question number 49 says, given below are two statements. Statement 1 is, a modified Hogland solution contains all known mineral elements needed for rapid plant growth. Yes. Statement 2 is, the concentration of these elements are set at the lowest possible concentrations. No, they are set as highest possible concentrations without producing. Yes, and thus maybe several orders of magnitude lower than, no, higher than. What's the point if it is lower than those found in the soil? What's the point of giving this Hogland solution then? So this one is not right. So what we have to do, we have to, in the light of above statements, choose the most appropriate answer. Both 1 and 2 are correct. No. Both 1 and 2 are incorrect. No. 1 is correct. 2 is incorrect. Okay. Mm, look at here. 1 is incorrect, but 2 is incorrect. Nice. Given below are two statements again about photosystem 1 and second. Question number 50. Uh, PS1 preferentially absorbs far red light of wavelength greater than 680. I think yes, because it is also called P700. So it absorbs in the far red region. PS1 produces a strong reductant, capable of reducing NADP positive and a weak oxidant. Yes, NADP positive is reduced to NADPH. So this statement seems correct. Statement 2 is, PS second preferentially absorbs red light of 680 nanometers. Yes, P680, and is driven very poorly by far red light. Okay. PS second produces a very strong oxidant capable of oxidizing water. Yes, absolutely. And a weaker reductant than the one produced by PS1. Right. So this is also correct. Both statements are correct. Statement 1 and 2 are correct. So option A. Coming to the question number 51. Given below are the statements about photoreceptors. Phototropins regulate phototropism, chloroplast movements, and stomatal opening. Right, absolutely. This one is right. Statement 2 says phytochrome absorbs red light and far red light most strongly. Okay, but also absorbs blue light and UVAP and mediate. Yes, this is also correct. Cryptochrome mediates several blue light responses, including suppression of hypocortical elongation, promotion of choroidal expansion, inhibition of petiole elongation, anthocyanin production, and circadian clock. This is also correct. Yes. Which of the following statement is correct? Okay, 1, 2, and 3. Now coming to the question number 52. What it says, during ABA mediated stomatal closure, membrane depolarization is caused by net influx of which of the following ions. So during ABA mediated stomatal closure, which happens under especially drought conditions, uh, membrane depolarization is caused by net influx of which of the following ions. So chloride, no. Chloride are actually pumped out, so this shouldn't be. Potassium, the entry of potassium is prohibited, and hydrogen, no, not much. And calcium, yes, calcium is pumped into the cytoplasm, so this should be calcium. Okay. 52's answer should be D. And moving on to the next question. The pigment molecules responsible for photosynthesis are located in they are located in thylakoid membrane of the chloroplast. Okay. Question number 54. So what it says that which of the statements about the C4 plants are not true. Okay, about C4 plants. The carbon dioxide compensation point is about 50 to 100 ppm. No, it's not. It's about 3 to 10 ppm in case of C4 plants. So this should be wrong. They have lower net assimilation at high temperature in comparison to C3 plants. Not, absolutely not, because they have higher net assimilation at high temperature, because C4 plant is, or C4 mechanism is an adaptation to the higher temperatures. Their bundle sheath cells contain many large sized chloroplasts. Yes. Fixation of carbon dioxide occurs by carboxylation of PEP, phosphoenol pyruvate, in the mesophyll cells. Yes, this is also correct, because these mesophyll cells have PEP carboxylase. PEP carboxylase. And then decarboxylation of C4 acids within the bundle sheath cells, and generation of carbon dioxide, which then enters in Calvin cycle. This is also correct. So 3, 4, 5 are correct, and 1 and 2 are incorrect. So incorrect we have to choose. Yes, so option A is the correct answer. Moving on to the next question. Question number 55. Which one of the following combinations represent the correct location and their associated key functions? Bowman's capsule, medulla of the kidney, ultrafiltration? No. Proximal convoluted tubule is in the cortex of the kidney? Yes. Regulated salt water organocellulose reabsorption? Yes. 55's answer is B. Moving on to the next. Which of the following is not a transcriptional activator? So option A is PTEB. PTEB is an elongation factor in eukaryotes. PTB is an elongation factor in eukaryotes. Zinc fingers, leucine zipper, helix, turn helix, or the motifs of transcriptional activators or transcriptional factors. Moving on to the next question, number 57. Given below are the steps of action of a water soluble hormone on its target cell. Okay, 1, 2, 3, 4, 5. What we have to do? Choose the option representing the correct order the action from below. Okay, we have to arrange these steps that happen in a soluble hormone signaling pathway. So the first one will be 3, 3. The hormone binds to a membrane receptor. So yes, this should be the first. Second should be this cancels out C and D. Let's look at 5. The hormone receptor complex activates G protein. Yes, right. So this is the second. I think A will be its correct answer. What about 1? 1 is adenyl cyclase. No, it happens after this activation of G protein. Okay. Question number 58. Function of enterogasterone is which is in the stomach, inhibition of bile flow, inhibition of gastric secretions, plus gastric mortality. Okay. Question number 59. During DNA replication events at the replication fork require various types of enzymes. Yes, having specialized functions, except DNA polymerase 3 is needed. DNA glyco. Yes, this one is not needed. So option B. The amount of oxygen dissolved in human plasma is, okay, it's around 2%. Okay. Which question number 61. Which of the following modifications of proteins is not post-translational in mammalian systems? Post-translational modifications of proteins. Palmitylation, yes. Glycosylation, yes. Phosphorylation, yes. Peptidylation, no. So this is the correct answer. Okay. Question number 62. In which year the International Botanical Congress adopted the International Code of Nomenclature for algae, fungi, and plants, and replaced the International Code of Botanical Nomenclature? In which Botanical Congress? In 2011. This one, which happened in Melbourne. The ICBN was changed to ICN. And yes, the and one important thing here you can take is that the algae is starts with small letter, so is fungi as well as plants, because These are not according to, these names are not according to the rules. Okay, so these are just layman language that we use for these groups of plants. Okay, so what is the current code? It is the Madrid code. Madrid code that came out in 2025, July 2025, which superseded this Shenzhen code. Okay, moving on to the next 63. Children with Patas syndrome have trisomy 13. Okay, okay, 64. Question number 64, which is in Drosophila melanogaster, the allele for yellow body color is Y, is located on the X chromosome. So this is located on the X chromosome. Yellow body color is recessive to the wild type body color. So this is recessive to the wild type body color. Uh, a yellow bodied male is crossed to a wild type heterozygous female. So you have a yellow bodied male which would be like this, is crossed to a heterozygous wild type. So the heterozygous wild type will be like this. When you cross them, and you will get which one of the following statements correctly represents observations of F2 progeny. So all females are expected to show wild type body color. No. All males are expected to show yellow body color. No. Um, I think this one will be the correct answer, because 50% of the males are expected to show yellow body color, because they will get one X from the female, which would be XY, and the other X would be XW. So I think half of the males are expected to show yellow body color. Yes. Okay. Moving on to the next question, question number 65. A transgenic plant was developed with a gene bar that was resistant to the herbicide Besta. Okay. The gene is integrated at a single locus and is present on only one of the pair of homologous chromosomes. So if you have a homologous chromosome, it is present on this bar gene is present on only one homologous chromosome, not on the other. So you have chromosome, okay, and these are the two homologous chromosomes. This transgenic plant was sulfid. Okay, so CB and C was sulfid. CBC. What percentage of the progeny is expected to show resistance to Besta? Okay, so what we will get? We will get CBC, CB, CB. Okay, mm-hmm. CC, B. Okay, CC. So the gene is present on 1, 2, and 3. 75% is the correct answer. Okay. Question number 66. Segregation of alleles can occur at anaphase 1 or at anaphase 2. Okay. In which one of the following organisms can first division segregation M1 and second division segregation pattern be distinguished? Okay, anaphase 1 or at anaphase 2. Yes, it could be due to crossing over happening in the anaphase 1. So yes, in ascomycetes, you may have heard about tetrad analysis, where you can easily distinguish between the first division segregation and second division segregation of allele, whether the alleles segregate in the first division. That is, we have a homologous pair, okay, and suppose we have any gene which has A allele, and on the other homologous pair it has small a. These will be separated in the meiosis 1, or they could separate if there is a combination between the daughter chromatids. It could separate in the meiosis 2. So this is easily indistinguishable in the ascomycetes. So which one of these belongs to ascomycetes here? Okay, Neurosporoprasa. Okay. Question number 67. In Orchigonity, which of the following plant groups are studied? Orchigonity means those plant groups which have orchigonia. Ulva, it's an algae, so you don't find orchigonia here. Rixia is a bryophyte, terrace, and pinus. Yes, option B is the correct answer. This is the logo of it is the logo of CBD, Convention on Biological Diversity. Question number 68. Which of these groups of plants produce seeds without fruit? So these are gymnosperms, obviously cycads. Question number 70. Sedges are common name of. Okay. Sedges are grass-like plants having triangular stem. As you may have heard that sedges have edges, rushes are round, grasses have nodes from top to the ground. Okay, so sedges have edges means they have triangular stem. They are grass-like. They belong to the family Cyperaceae. Okay, moving on to the next question, 71. Which of the following is not a larva of Echinoderma? Bipineria, yes. Veliger, no. This is also a larva of Echinoderm, and so is this Doliolaria. So Veliger is not the larva. Moving on to the next, 72. Which one of the following is not a characteristic feature of Urochordata? Pharyngeal gill silts. Okay, they do not have this cranium or brain box, distinct cranium or brain box. 72. Moving on to the next, 73. Match the disease list one with pathogen. Cruz Feltus Jacob. It's a prion disease. Okay. Pneumocytes. I'm not sure. You have rabies is a viral disease. So P3. Okay. Option B is the correct. Okay. Now moving on to the 74th question. Which of the following acts as a test organism in the procedure of phenol coefficient method? Phenol coefficient method, which is the standard organism, it is Staphylococcus aureus. Staphylococcus aureus, or sometimes they use Salmonella typhi. This method, this phenol coefficient method, is used for testing the efficiency of different disinfectants. Okay, and they use phenol as the standard. Moving on to the next question, 75. Cryptococcosis is a disease caused by fungus, so mycotic infection. Moving on to the 76. In geological timescale, currently we are in Phanerozoic Aeon, Cenozoic era, and in Quaternary period, yes, and Holocene epoch, absolutely. 76's answer is A. Then 77, an aquifer. Okay, an aquifer is a rock formation which stores water, yes, in the pores, so it's porous and essentially permeable. Okay. 78. Question number 78. In the family Asteraceae, the pappus is often persistent fringe of scaly, bristly, hairy appendage. Yes, it's a persistent calyx. So A is the right answer. Moving on to the 79th question. Match the following pathogens. List one. Okay. Match the following pathogens and diseases given below. I thought we already did this. So, anyway, albigo candida causes white rust of crucifers, Phytophthora infestans, salade blight, Culiotrichum fulcatum, red rot of sugarcane, Alternaria solanae. Okay. So, P3 again. Q4. Yes. So, option C. Question number 80. Which of the following is or correct? Numerical taxonomy aims at determining phenetic relationships between organisms or taxa. Yes, this sounds correct. APG system of classification is established by taking the information from recent molecular studies. This also looks correct. Phylogenetic classification is constructed in such a way that all the descendants of a common ancestor should be placed in monophyletism. Yes, this also looks correct. So. Which of the options represents all correct options? So I think only 1, 2, and 3. We have only 3 options, so it should be this option D. Question number 81. Which of the following is the first national park of India? Oh, such a tough question. Jim Corbett National Park. Okay. Species A, question number 82. Species A produces many seeds smaller in size, while species B produces few seeds bigger in size. Select method followed by species. Okay. Smaller in size, many seeds. So R and K selection. Yes. Question number 83. Which of the following combination of Nutrients follow sedimentary pattern of biogeochemical cycling. Carbon, no. Sulfur, yes. Phosphorus, you have nitrogen. Nitrogen, carbon, no. Moving on to the question number 84. Which of the following statements describes the characteristic of a keystone species? The species which dominate in the ecosystem, no. Keystone species are not dominant. Necessarily, they are not dominant. The species those occur outside their natural range, no, not at all. The species which has low abundance and biomass in the community, but it has large ramifying effect on the community structure, yes. Okay. C should be the correct. The species with its rare occurrence and restricted distribution and no effect, no. Moving on to the next question number 85. Which of the following statements is not correct? Pyramid of biomass in a grassland is upright. It's correct. Pyramid of biomass in a pond is inverted. Yes, it's also correct. Pyramid of energy in any is always inverted. No, it is always upright. So C should be the correct answer. Statement is wrong. So, but the but it is the correct answer to this question. Moving on to 86. India is one of the 17 mega diverse countries. What is a mega diverse country? A country should have around 1500 species as endemic, and it has to have marine biodiversity or marine ecosystem in its border. Question number 87, which says that given below are two statements regarding the ecosystem stability. Statement one goes like, the tropical rainforest ecosystems are said to be highly stable and resistant because they have strong resilience. Once greatly perturbed, they may quickly return to the original state. Yes, statement one is correct. Mycorrhizal symbiotes act as positive feedbacks by amplifying the processes that increase the ecosystem stability and self-sustaining ecosystem processes. Right. So both statements are correct. Okay, option A. Moving on to the next question. The following escargot represents a logistic population growth. Which of the following marks as option A to D on the on the y-axis refers to the carrying capacity? Okay, here is the carrying capacity P. P should be the answer. Question number 89. Capture mark recapture method is used to estimate, yes, populations. So in an experiment, 250 grasshoppers were captured. You have 250 grasshoppers were captured on day one from a field in Jammu University. They were marked and released. On the second day, 175. On the second day, 175 grasshoppers were captured from the same field. Okay, this was day one, and this is day two. Of the recaptured grasshopper, 70 were marked. What is the estimated population? Okay, you can do this by 250 out of x equals 70 out of 175. Then you can simply cross multiply these, and the answer is 89's answer is 625. Which type of protected, question number 90, which type of protected area conservation introduced after the amendment to the Wildlife Act 1972 in 2002 is wildlife sanctuary? Natural selection can alter, question number 91, the frequency distribution of heritable traits in different ways depending on which phenotypes are favored. In one of the selection modes, the birth weights of most human babies lie between 3 to 4 kg. Okay, babies who are either much smaller or much larger suffer higher mortality rates. This is an example of um, stabilizing selection, I think it is. Okay, so moving on to the next, which is question number 92. A population of squirrels which could interbreed was split into, so you have a population which was split into two groups by a waterway in between because of a man-made waterway. Okay, this led to the reproductive isolation of and formation of new species. This is an example, allopatric speciation. Allopatric means, allo means different, patric means fatherland. So these are those species which arise from two different geographical area. Question number 93. Which of the following statements is correct about telomerase? Telomerase has 5' 3' DNA dependent. No. It's RNA dependent DNA polymerase. Okay, so question number 93's option is C. Question number 94. Which of the following assumptions support the Hardy-Weinberg equilibrium? It's presence of natural selection, no random matting. Okay, a Hardy-Weinberg equilibrium is an ideal system where you have random mating, no natural selection, no genetic drift. Okay, question number 95. Which one of the following components is necessary for screening of recombinant plasmid molecules? A gene like beta lactamase, beta galactosidase, multiple. A gene like beta lactamase. Beta lactamase is ampicillin resistance gene. Okay, which which hydrolyzes the beta lactam ring of ampicillin and thus provides resistance to the gene bearer. 96. What is the generation time of a bacterial culture that grows from 10 raised power 2 to 13 into 10 raised power 3 in 210 minutes? Okay. There are a few ways that you can use to solve this question. Let me select the color here. Okay. Uh, Generation time, it's the time between doubling. Okay, doubling of bacteria. So from 1 to 2, how much time bacteria takes. There is a formula which is G equals T divided by 3.3 log of nt by n0. But in this case you have to know the log of what comes here. That is, t is the time period, which is 210, and then 3 divided by 3.3 log of nt is the number of individuals at time t, which is 13 into 10 raised power 3, which is roughly 13,000. Log of 13,000 divided by n0, so 100. So this will get cancelled. Log of 130, which is roughly around 2.2. So 3.3 into 2.2. Okay, it will become 210 divided by some 7.2, and that comes around 730 30 minutes. There is another way, which is like nt equals n0 into 2 raised power t over g. Then it becomes nt over n0 equals 2 raised power t by g. Nt over n0 is 13,000, okay, divided by 100, and it will become 130. So 130 equals 2 raised power time interval is 210 divided by g. G is the generation time. So what should be the power of 2 so that it becomes 130? Okay, it should be 2 raised to the power 7. As I have done the calculations, it should be 2 raised to the power 7. But how will 7 come here? You have to kind of go backwards. 7 will come here by when you divide this 210 by 30. Okay, so it will be like 2 raised power 210 divided by 30. So g is the generation time here. Okay, and this is a short neat trick that you can use. So this is the main formula. Okay. Okay, moving on to the 97th question. Match the list 1 with the list 2. So toluidine blue is used for nucleic acids. Lactophenol, yes, it is used for fungal hyphae. Sudan 3 is used for um, lipids, and this is used for proteins. So toluidine blue RNA P3, okay, P3 here. Lactophenol 4, Q4. Okay, so this is the correct option C. 98 question. Which species concept is would you apply to both sexual and asexual species? So it's the morphological yes, ecological yes, no, morphological yes. So option D. Okay, question number 99. Consider the following statement. Statement 1. Chimeric antigen receptor CAR. T cell therapy has emerged as a novel therapeutic cell engineering which polymorphonuclear cells derived from patients are engineered in vitro. Yes, this is correct. The use of CRT cell therapy has been successful for treatment of acute lympho. Yes, this is also correct. So both statement one and two are correct. 100 chromatographic technique, wherein the size of a molecule is the primary criteria for separation of biomolecules, is called. Ah, we have already solved this question. It's called gel filtration chromatography, which is based on molecular size. Okay.